Hi everybody, this is Dr. A, and this video is going to be an overview of the blood as part of the chapter on blood, lymphatic, and immune system. Alright, so blood transports substances throughout the body. These substances can be attached to red cells or they can be dissolved in plasma. For example, oxygen is attached to the red cell, and we do need oxygen delivered to all of our cells, but substances that might be dissolved in plasma could be some of your vitamins, amino acids, and other electrolytes and other substances like such. The white blood cells fight infections and disease. The platelets initiate blood clotting processes. And the red cells, they carry oxygen. So the components of blood are the blood cells, also known as the formed elements, and that's because platelets aren't actually cells, they're cell fragments. But anyway, the formed elements, if you will, for the blood are the erythrocytes, which are the red cells, the platelets, which are cell fragments, and the leukocytes, which are white cells, and then the plasma, which is the liquid that these cells float around in. The average adult has about 5 liters of blood. If you need to picture that, you can picture a 2-liter soda bottle, and that would be 2.5 of those. Blood is a mixture of cells floating in the watery plasma, and there are 3 types of blood cells or formed elements: erythrocytes, red cells, leukocytes, the white blood cells, and platelets, which are cell fragments. The blood cells are produced in the bone marrow, and so are the platelets. And the platelets are fragments of a cell called a megakaryocyte. The plasma, about 55% of the blood is plasma, and so therefore about 45% will be the formed elements or the cells. And plasma itself is 90 to 92% water, and 8 to 10% dissolved substances. One of the predominant dissolved substances in plasma is protein, and so the protein that is in plasma, about half of it is albumin, and so albumin transports fatty substances, but it can also ferry drugs and other things. The reason why you need albumin to transport a fatty substance is because fatty substances aren't water soluble, and plasma is mostly water. And so if you put the fatty substances and you attach them to a protein, it makes them water soluble and therefore transportable in the blood. Your gamma globulins are proteins that act as antibodies. They have generally a Y-shaped. And the fibrinogen is another protein that is a clotting protein. So once activated, it can become fibrin. So let's start with the erythrocytes, or the red cells. They are enucleated biconcave discs. What that means is when they were formed in the bone marrow, the nucleus was removed. It kind of spits out its nucleus before it leaves the bone marrow and enters the circulation. So a red cell does not have a nucleus. And because it doesn't have a nucleus, it can assume this biconcave disc. So with that middle depression that you can see there, right here in the cell, it's on both sides. And that form makes it very flexible. It makes it able to kind of squeeze through the capillaries, which are often just the width of one red cell. Adults have about 35 trillion red blood cells. And hemoglobin gives these cells their red color. Hemoglobin is a pigment that contains iron, and it is responsible for the transporting of the oxygen. So the oxygen binds on the iron component, which is hemoglobin, and that's how it's transported in red cells. An average red cell will live 120 days, and then they are removed from circulation by the liver and the spleen, and recycled, like the iron is recycled, and the amino acids are broken away from the hemoglobin portion, or the globin portion of the hemoglobin. And so some of that is reused, some of that is discarded. Next, we have the leukocytes. The leukocytes are white blood cells. They are spherical, so they're round with a large nucleus. White blood cells provide protection against pathogens, all kinds of pathogens, viruses, bacteria, parasites, fungi. There are two big types of leukocytes simply by what they look like. So we have granulocytes. They have granules in their cytoplasm, and the agranulocytes do not have granules in their cytoplasm. So those are two big categories. And your granulocytes are going to be your neutrophils, eosinophils, and basophils, and your agranulocytes are going to be your lymphocytes and your monocytes. So let's look at the three types of granulocytes. So basophils, they release histamine and heparin to damage tissue. They have really dark purple staining granules. So the basophil is represented here. They're not a majority of your white blood cells. They're only about probably 2% on average of your white cells. Same for the eosinophils. They are only about 2 to maybe 5% of your white cells. These two are not found in large numbers. Eosinophils are made to destroy parasites, and they will also increase during allergic reactions. So if you have things like hay fever or just an allergic reaction to a drug or something like that, your amount of eosinophils is going to go up. If you have like chronic allergies like hay fever, you're likely to have high basophils and high eosinophils. But when we talk about high, we're not talking about like huge numbers. So your basophil count might go from, instead of being like 1 to 2%, it may go up to like 10%. Eosinophils, eosino means rosy red, they have these orangey red granules in their cytoplasm, and so they're really pretty to look at when you see them. And then you have your neutrophils. Your neutrophils, they engulf foreign material like bacteria and also bits and pieces of dead cells via phagocytosis, so it can eat things. And it has a lobe nucleus, has multi-lobes, so the nucleus takes on this odd shape here. Same with the eosinophil, but the eosinophil usually doesn't have as many lobes as the neutrophil. And the neutrophil has a few light purple granules, and this is one of the most abundant cells, white cell, in your blood. So it's on a normal day, you should have about 60 to 65% of your white cells that are neutrophils. If you get a bacterial infection, your neutrophil percentage will go up, and it can go up to the 80% and upper 90% of the white cells if your body is fighting a bacterial infection. There are two types of the agranulocytes, so we're going to look at the monocytes and the lymphocytes. So the monocytes are represented here. They're the biggest of the white cells, and they're made to engulf foreign and damaged cells also via phagocytosis. They are not picky. They will eat anything that doesn't belong, and they help coordinate the immune response with the lymphocytes. And then lymphocytes provide protection during the immune response. One of the things that lymphocytes do is to produce antibodies. They are much smaller than the monocytes, and they are a little bit larger than a red cell, and they have a large nucleus in comparison to the amount of cytoplasm that they have. FYI, also a monocyte that has left the blood circulation is called a macrophage. And next we have the platelets. So platelets are the smallest formed element of the blood. They are plate-like fragments of a larger cell called a megakaryocyte, and they've broken and been pinched off, and that's how they enter the circulation. They are critical to blood clotting, so they get activated when blood vessels are damaged or cut, and when they activate, they put little extensions out and grab each other, and they agglutinate at the site of the damage. So that means they link up to each other, and they trap red cells and white cells, and they release substances that contribute to the clot formation. So they are critical to stopping bleeding. So in this electron micrograph, you see a red cell right here, a platelet right here, and then a white cell right here. This is most likely a lymphocyte due to its relative size to the red cell. So lastly, we're going to talk about blood typing. So each person's blood is different from the blood of others, and you have marker proteins on the surface of your erythrocytes that cause these differences. These are genetically determined, and therefore we have to do a blood type before being able to give blood to somebody, and we actually have also to do a cross match. So we type the blood of the recipient, the person needing blood. We are generally do an antibody screen, so we check for the presence of different antibodies, and then we also make sure that their blood is compatible with the units of blood that we have selected to donate them to them. So the ABO and RH systems are key to blood typing. They are not the only system, but this is the one we're going to talk about because it's the principal one that you have to get this one right for sure. So there are two possible red blood cell markers. There are A and B, and they are actually glycoproteins that are on the surface of the red cell. So their little markers are sticking up on the surface of the red cell. So if a blood has the A marker, then it's type A blood. And through a process that we don't yet fully understand, after birth, if you have type A, if you have an A marker, you will make anti-B antibodies. You'll start making anti-B antibodies. The anti-B antibodies that float around in a person that has type A will attack the B marker, right? And so a person that is type A could not receive B blood, or they could not receive AB blood because it would be deadly to them. Blood with the B marker is type B blood. And so they produce anti-A antibodies, and those antibodies would attack type A blood. So if a person was B, they could not be given type A blood or type AB blood because it would also kill them. But there are some other possible combinations. So you can also have both markers. You can have the marker of A and the marker of B. And so that is an AB blood type. And because they have both markers, they will not produce any kind of antibodies. Because they have A, they won't produce anti-B. And because they have B, they will not produce anti-A. So they have zero AB antibodies anyway. So they will not attack any other blood type because they have both markers. And as such, they are the universal recipient for packed red cells. So we can give them type A, type B, type AB, and type O, which we're going to talk about here in just a second. So if a blood does not have any of the markers on its surface, so it does not have A, it does not have B, then it's a type O. Okay, and unfortunately then type O will produce both anti-A and anti-B antibodies because they do not have those markers. So you always make antibodies to what you don't have. And therefore it will attack all other blood types. And so type O can only receive type O. It cannot receive type A because it would react with the anti-A antibodies. It cannot receive type B because it would react with the anti-B antibodies. And it cannot receive AB because the antibodies would attack both A and the B antigens, markers on the surface of those red cells. But its advantage is because it does not have any kind of markers, it is not attacked by antibodies in other blood types, and therefore it's a universal donor. So a person that has type A can receive type O. A person that has type B can receive type O. A person that's type AB can receive type O, and that's why they are the universal donor. So then we need to also put in the RH factor, because the RH factor is also really important. And so there are two possibilities with RH factor. You are either RH positive or RH negative. If you are RH positive, you will never make any anti-RH on antibodies. The RH marker is considered the D antigen, so the D marker, big D. And so RH positive is big D, And if you are Rh negative, you are little d. And if you are Rh negative, you do have the possibility to make anti-Rh antibodies. But you would only do so if you are exposed to the Rh antigen in some way. Some of the ways that this can happen are through a transfusion of an Rh positive blood into an Rh negative person, which would be very rare for that to happen. And then the other way would be through pregnancy and childbirth if an Rh negative mom is carrying an Rh positive child. So Rh positive blood has no antibodies, no anti-Rh antibodies, and so it cannot attack any Rh negative blood type. If you are Rh positive, you could receive Rh positive and Rh negative blood. And so that adds a layer of complications, because then you combine that with the A, B, and O blood types, and then you have the 8 blood types, which are A positive, A negative, B positive, B negative, AB positive, AB negative, O positive, and O negative. Okay, so next we have to understand that if you are Rh negative, you could potentially make an anti-Rh antibody or an anti-D antibody, and then you would attack Rh positive blood. So what we do is for patients that are Rh negative, we only give them Rh negative blood. So then when you combine it all together, you have all of these blood types. So let's think about this. So we have, let's see, AB positive. So AB positive has the A marker here, the B marker, and the Rh marker. Okay. It is truly the universal recipient because it will not have anti-A, it will not have anti-B, and it will not have anti-Rh. So it literally can get any of these other blood types without any kind of problem. All right, next you have AB negative. So AB negative has the A marker and the B marker, but it does not have the Rh marker, which means that it can receive all of the other blood types that are Rh negative. So AB negative can receive A negative, B negative, and O negative. All right, so next we have our A blood type. So A positive has the A marker and the Rh marker, and therefore it doesn't have B, so it'll have anti-B. And so a person that is A can receive A blood, that's A positive, O positive blood. They could also receive A negative blood, because remember you can give Rh negative blood to Rh positive person, and they can also receive O negative blood. Okay, so then we have A negative. So A negative has an A marker but not the Rh marker. So they would have anti-B, but they might also potentially have an anti-Rh antibody. And therefore, a person that's A negative can receive A negative blood, and they can receive O negative blood, but not any other type. So you cannot really give them any A positive or O positive, because they might then have the Rh antibody. And next is B positive and B negative. So B positive has the B marker and the Rh marker, and therefore it will have anti-A antibodies. And so you can give B positive and B negative blood to a B positive patient. You can also give them O positive or O negative, but you cannot give them anything that has A. You cannot give them any of those types. And then with the B negative, they have the B marker, so they'll have anti-A antibodies and potentially anti-Rh antibodies, so they can only receive B negative blood cells or O negative blood cells. They cannot receive the Rh positive ones. And then we have O positive. It just has a Rh marker on the surface of the cells, and therefore it can make anti-A and anti-B antibodies, and so it can only receive O positive or O negative, because remember it can receive Rh negative blood. It cannot receive any of the others because they would all react with the antibodies that it has. And then this one is truly the universal donor because it does not have an A marker, a B marker, or an Rh marker. Therefore, it's compatible with all the other blood types. Unfortunately, because it can have anti-A, anti-B, and anti-Rh, the only blood type that O negative can receive is O negative. All right, so I hope that clarifies the blood types for you guys, and that you get this good baseline, and that this is a good review. So that wraps up our video on blood.